Our last speaker of the day is Rebekah Beyea, who will be talking to us about long-read RNA sequencing reveals differential mRNA expression and transcript usage in AKI and PKD mouse models.
Yes, thank you. So this summer I analyzed RNA sequencing data in our mouse models for acute kidney injury and polycystic kidney disease. So let me start with some background information. The genome is the complete set of genes in an organism, while the transcriptome is the complete set of transcripts in an organism. And after this process of transcription from gene into transcript, there's this mechanism called alternative splicing that allows multiple different transcripts to be produced from the same gene, depending on which exons have been included. And so what we did is long-read RNA sequencing, which is able to capture full-length mRNA transcripts, and we map these reads to the transcriptome to determine which transcripts are being used in different conditions. So analyses we can do from there include differential gene expression, which looks at the changes in the expression of a gene between different conditions. So for example, this shows that gene A is expressed more in the first condition than the second. Differential transcript expression looks at the same idea but with these transcripts. And then differential transcript usage looks at the changes in transcript expression but in proportion to other transcripts from the same gene. So let me give some background on acute kidney injury and polycystic kidney disease, which both frequently lead to kidney failure. Acute kidney injury is defined as a sudden reduction in the level of kidney function, and those that recover are at a much higher chance for developing chronic kidney disease. And one form of this is polycystic kidney disease. The autosomal dominant form is one of the most common genetic diseases, and it's caused usually by mutations in the PKD1 or PKD2 genes. And this disease is characterized by fluid-filled cysts that cause the kidneys to enlarge and their tubules to dilate. So in our study, we modeled both of these. For AKI, we use the Beyea surgery, which inhibits the blood flow to the kidneys and causing this acute injury. For PKD, we use the RCRC model, which is a mutation in the PKD1 gene to cause a mild form of PKD based off of the human patient variant model. So we have 4 samples, one with AKI, one with PKD, one with both, and one with neither. And our goal was to identify differential expression and transcript usage between our 4 models. So to do this, we isolated RNA from those kidneys, did RNA sequencing on an Oxford nanopore crude ion, and then did gene and transcript analysis from there. So in all of our downstream analyses, our AKI models are both of the mice with the Beyea surgeries, and our controls both have the sham surgery. For PKD, both mice have the RCRC mutation, and wild type are our controls. So the first analysis I did was on differential transcript usage. And those genes that we found with differential transcript usage we called switching genes. So we compared AKI to control and PKD to control and found 5,000 and 4,400 switching genes respectively, and a 52% overlap between those. Then we looked into the literature to find genes that are already known to play a role in AKI or PKD, and I look into a couple of those more specifically. So one of those is VCAM1, which codes for the vascular cell adhesion molecule 1. And these are, in this switch plot, you can see the 2 different transcripts that can be produced from this gene, and they are color-coded based on known protein domains. And our analyses showed an increase in the gene expression of VCAM1 during AKI, which is our Beyea model in gray, compared to our control sham model in black. And literature supports this increase in the gene expression due to the expected upregulation of cell adhesion molecules following damage, as it promotes leukocyte infiltration to aid in healing by inflammation. In addition, we can also see differential transcript expression and transcript usage. The second gene I looked at was PKD1, which codes for polycystin-1, a cell surface membrane receptor. And here we can see the 3 different transcripts that can be produced from this gene. And again, they're color-coded by known protein domains. Our analyses showed a decrease in the level of gene expression of PKD1 during PKD, which is our RCRC model in black, compared to our control wild-type model in gray. Because the kidneys rely on this balance of polycystin-1 and another protein, polycystin-2, for normal function and development, literature suggests that both an increase or a decrease in this gene expression can cause clinical features of PKD, with more severe imbalances causing more rapid cyst formation. Again, we can also see differential transcript expression and transcript usage in these graphs. So to conclude, we did bulk long-read RNA sequencing of mice kidney tissue to look at differential expression and transcript usage between our models for AKI and PKD. We identified genes with differential transcript usage that we called switching genes, and a 52% overlap between these conditions. We also investigated 2 genes, VCAM1 and PKD1 further, expression results align with current literature. As for future directions, this is a pilot study and provides preliminary data justifying the expansion of these analyses to more samples. And finding some of these alternatively spliced transcript patterns can help with finding insight into the disease as well as with future drug development. In addition to the analyses that we have done, we can investigate specific switching genes further along with additional factors. And finally, an additional technique to use would be single-cell RNA sequencing, which would allow us to look at patterns in individual cell types of the kidney. And so I'd like to thank the Lisane Lab and UAB KURE for this experience, as well as the George Yoder and Ruck Labs for providing our samples. All right, any questions? Yes, right here.
Can you go back to the analysis with the graphs? I think so. Yeah. Can you give a example of the function of those proteins colored and mixed?
The protein domains. One example would be that, like the top light blue one, the lectin C, is a carbohydrate binding domain. So since this is a cell surface membrane receptor, it allows carbohydrates to bind to the receptor. Yes, very good.
Maybe a mean question, but do you know if polycystin 1 and 2 are expressed in a circadian pattern?
I do not know that. That's definitely, I could look in the literature for that, but we did not look at circadian rhythm.
There's a lot of genes in the kidney.